But got it. Okay, but a lot of the focus on sleep and Alzheimer's has been focused on sort of global sleep aspects, but I really want to nail down into some of the local sleep features that I've been looking at and talk about kind of the different ways we can conceive of this link, because you can conceive of this link from the angle of sleep disturbances facilitating disruptions that lead to Alzheimer's pathophysiology or dementia pathophysiology that leads to cognitive decline. But you can also see it from the angle of Alzheimer's pathophysiology disrupting sleep features that are relevant for cognitive function. So there's different ways in which sleep is intertwined with this relationship. So I'll talk about a little bit of that, but also I'll talk about aging as well and how it's impacted. And really a lot of this question to me comes from this, basic need to understand the variability in trajectories of cognition across the lifespan. So we know that as time passes, some people may decline very little or some, but others have a more precipitous decline, with the extreme examples of that being in different forms of dementia like Alzheimer's disease. And a lot of the work in that space had been focused on, what if I take someone who's really declined in their late stages of dementia or the beginning stages of the dementia, let's try to find some kind of treatment or find some intervention to try to rescue their cognition. And for the most part, those have all failed. And this has led to a big crisis in Alzheimer's research in general. There's a lot of failed clinical trials. And this has led to kind of this view over the last decade or so that we really need to be focusing earlier, because it's dementia and cognitive decline associated with it are caused by a cascade of biological events that result in synaptic degeneration throughout the brain. And then once you lose those synapses, once you have system-wide degeneration, maybe it's too early to treat the pathology. And so a lot of focus has been trying to, kind of revisit preclinical stages, prodromal stages like mild cognitive impairment, or preclinical stages even earlier when pathology starts to begin to be deposited, and try to see if you can predict or separate, these people on different trajectories of cognition. And one of the things that myself and others in the field, like Masood, have been trying to do is try to see, well, is sleep a critical factor in these cognitive trajectories, and try to understand what role sleep might play in that decline, and can it be a source of targeted intervention or early identification, so you can have early interventions at this stage. And why do we think of this? Well, we think of this because there is a substantial literature now showing that that sleep plays a critical role in memory retention or memory consolidation or memory encoding even, such that in this literature, if you have sleep, it aids in the consolidation offline after initial learning, that aids recall or retention minutes to hours to years later. And cueing memory this has been demonstrated causally by a variety of different approaches, including cueing memory during sleep to stimulate specific oscillations to enhance memory, or to reactivate specific memories, and showing that can be enhanced by sleep. Sleep deprivation and disrupting specific oscillations during sleep also impairs memory. And so one of the core questions is, what are the mechanisms of this? What are the features of this? And there's been a lot of work that's kind of drilled down and focused on this. And it really comes down to the fact that sleep is defined by local oscillatory activity in many ways. And there's different features across wake and sleep brain states that seem to be relevant for these phenomena, which I'll get into in the next slide. But essentially, if you're looking at sleep and you're looking at wake, some of the defining features of wake, for example, are a local expression of alpha activity in occipital cortex. It really defines kind of their drowsy, eyes closed wakefulness. But in non-REM sleep, sort of that once you start getting into deeper stages like stage 2 sleep, you start seeing the expression of signature oscillations like sleep spindles, which have a very specific characteristic topographical pattern, whereby in slower examples of spindles tend to be more frontally sourced or frontally expressed, whereas faster ones are more parietal centrally expressed. And then you have slow waves, a dominant defining feature of sleep, which tends to be more frontally expressed. And these are sourced to different cortical areas or subcortical areas. And REM sleep, which looks, kind of a lot more like wakefulness, also has local oscillatory features, including theta as well as beta and gamma, which are expressed in sort of locally dominant ways. And there are three different signatures that really are focused on in the literature of aging and Alzheimer's disease. And that is expression of spindles, expression of slow waves, their relationship with each other, expression of ripples too, but you don't really typically measure them on the scalp, but also just the characteristic features of REM sleep itself and how these may be linked to different aspects. And in terms of sort of memory theory, there are different cortical systems that interact with each other. This is a figure from an older paper from one of the more famous sleep researchers in the field, who is from your country, Jan Born, who's done some elegant work in this area across humans and animal studies. But this identifies oscillations, slow oscillations, sleep spindles, and hippocampal ripples, and how they all phase lock to each other and across different cortical networks involving the hippocampus, the thalamus, and the cortex. And this may facilitate essentially communication and replay and communication between the cortex and hippocampus, facilitating the cementing of memories into the brain long term. But in addition to that, there's characteristic neural circuitry that supports how REM sleep is expressed, both the theta rhythm in the hippocampus as well as the desynchrony we see in REM sleep. And this is essentially supported by glutamatergic and cholinergic inputs in the basal forebrain, the brainstem, hypothalamus, in interacting together to then facilitate the cortex's functioning. And both of these are thought to be supportive of memory consolidation. And so trying to understand the roles of these neural systems has been sort of focus of a lot of work. And there are three different theories. There's other theories as well, but here's three examples of theories that people posit of how sleep at a local level supports memory consolidation, or how quantitative aspects of sleep support memory consolidation. One is the classic active systems consolidation hypothesis, whereby you have slow oscillations, spindles, and ripples phase lock to each other in a very specific relationship to allow communication between the hippocampus and the cortex. And it's thought that this triple coupling, triggers replay during ripples that then facilitate how the cortex and neuroplasticity in the cortex selectively activates and changes synaptic organization. Another is the synaptic homeostasis hypothesis that suggests that as learning potentiates the brain during wakefulness, then sleep must act to homeostatically balance that to manage metabolic demands in the brain. And this process is thought to induce widespread LTD-like synaptic changes, eliminating weaker synapses, preserving stronger synapses, and then improving signal to noise. And these two theories have been talked about a lot and debated a lot in the literature. They're not necessarily mutually exclusive, but some of their predictions are very specific. For example, it's the slow oscillation, the less than 1 hertz frequency slow waves in the active systems consolidation hypothesis that is relevant for memory actively being enhanced, whereas in synaptic and shy, it's delta activity predominantly, or just slow waves across all frequencies that actually induce more LTD style changes. Another theory is that it's actually the interaction between REM sleep and non-REM sleep that matters. And this is some really nice work from NYU showing that memory causes specific select changes in neural systems, in particular certain specific branches on specific neurons, dendritic branches of neurons, that then form labile synapses during non-REM sleep, and then REM sleep acts to eliminate some and stabilize others. And this back and forth between the two is thought to be necessary. The physiological signatures of this phenomenon I don't think are as clear yet, but this process is thought to be one of the ways in which the two systems interact, and such that synapses are remodeled across these different stages. And these three distinct hypotheses of how different sleep features or sleep stages may support memory consolidation. And I said they're not necessarily incompatible, but specific deficits and specific sleep features would give us a sense of which of these theories is more supported by the data. And I think using aging and dementia, see how these different features are disrupted and how that relates to memory changes, can give us some insight into these phenomena. One thing I'd like to end with before this sort of the memory and sleep overview is that there's also been some really elegant optogenetic studies in rodents where they've actually manipulated different forms of slow waves, these slow oscillation, less than 1 hertz frequency slow waves versus these delta faster 1 to 4.5 hertz frequency slow waves. And they've showed that when you do that, when you disrupt the slow oscillation, it disrupts coupling between that and spindles, and it disrupts memory performance as well. And if you disrupt delta waves, you see an increase in slow oscillation spindle coupling and an enhancement of memory actually. And this is sort of interpreted to think that there might be different memory related functions if you're looking at the slow oscillation versus the delta wave. And this has been something that people have been talking about for a number of years now. And this finding just supported that hypothesis. But this has led to the kind of a focus on less than 1 hertz slow waves. Kind of already talked about this. Okay, so this might mean that that depending on which features of even waveforms you're looking at, whether it's slow or fast sleep spindles or slow or fast slow waves, you might have different neuroplastic functions or neuroplastic features associated with them, and different consequences when you disrupt different forms of them. So why is this relevant for aging? Why is this relevant for Alzheimer's disease? Well, aging and Alzheimer's disease have very characteristic changes that are associated with them in sleep and how it's expressed. Of course, this is probably one of the most widely shown slide in all of sleep research. I think probably it's been shown dozens, if not millions of times around the world in the last couple of decades. But this is from a classic meta-analysis that sort of from Hoy, and it really shows that across the lifespan, you have very dramatic changes in sleep, whereby if you look at time in bed, which is the addition of all of these bars together, and you break down the wakefulness periods, the sleep latency on the top, the wake after sleep onset below here, so this time spent awake during that time in bed. And then you have the breakdown in REM sleep, slow wave sleep, stage 2 and stage 1. And what's pretty clear is that across the adult aging lifespan, even with similar amounts of time in bed, there are 2 characteristic changes that you really see. One is wake after sleep onset increases. You get more fragmentation in your sleep as you get older. And slow wave sleep decreases. And if you look at your 20s to your 70s, you lose about 50% of your slow wave sleep in that time. So why is that happening? Oh, well, that's something that is being actively investigated. In addition to changes in architecture, we also see changes in oscillations. This is highly reproduced across studies for many decades now. So slow waves, for example, show a dramatic deficit. Here showing topo plots where nodes are on top, ears on either side. In this case, the whiter colors are more intense wave activity. And on the right, the whiter colors, brighter colors indicate more density, more incidence, or more of slow waves themselves. And this is young adults, older adults are on the same scale. You can see there's a dramatic difference. And this rainbow plot shows the subtraction between the two, whereby blue or cooler colors represent a bigger, the bigger difference between young and old, such that older have fewer than young. And so you see a dramatic age-related change over frontal cortex and slow waves. And when you look at fast spindles and slow spindles, even though they're sourced in different areas of the head, typically, you see the same kind of age effect predominantly over frontal cortex. You see a loss of spindles as well. So there's selective changes in expression of oscillations as well as architectural changes in sleep stages as well. And the big question that we've tried to address is why? Why are these changes happening? And what are the consequences of those changes? And so the first example I'll talk about is really just before we get into oscillations, why does sleep become more fragmented? And here's a study from Cliff Saper's group where they looked at histopathology of people they had actigraphy in a couple years before death. And they looked at the hypothalamus, and they identified an area that they thought of as a human homologue to the ventrolateral preoptic nucleus. In this case, it was the intermediate nucleus. And they looked for the staining of gallon neurons, and they said, okay, this is one of the areas, and these are the types of neurons that have been shown to support the expression of sustained, continuous non-REM sleep or slow wave sleep. And so theoretically, there should be deficits in this if you have more fragmentation and loss of sleep. And indeed, that's what they found. What you can see here on the horizontal axis is the number of inhibitory galanin neurons. And on the vertical axis is their measure of fragmentation, where higher numbers means more fragmentation. And you see that there's this inverted relationship in this relatively small sample, whereby the more of these galanin neurons you have, the less fragmentation there is. And the important thing for this, for relevance for Alzheimer's disease, is that they did have Alzheimer's disease patients in the sample, and those had the fewest neurons and the most fragmentation. So it wasn't the relationship was different once you got dementia, it was an exaggerated feature. So this suggests that some of the fragmentation is actually degeneration of sleep promoting centers themselves that occur with aging and dementia. Okay. So we also, and others also looked at the expression of slow wave activity. One of our very old papers at this point, we show that it was actually changes in the structure of the prefrontal cortex that was related both in young and older adults. The more volume, the more in the gray matter cortex, sorry, this is going on its own. The more, the less intense slow wave activity was, or sorry, the more intense slow wave activity was. And another group, Julie Carrier's group in Canada, basically showed also that changes in this medial prefrontal area, but also in lateral frontal areas and temporal areas and parietal areas were associated with the density of slow wave expression as well, suggesting that aging, age-related changes in cortical thickness and cortical volume were impacting slow wave expression, particularly in the frontal cortex. We tried to look at the functional consequence of this and showed, in fact, that retention of word pairs memory was reduced in older people relative to younger people. That's they showed a bigger drop in memory, and this was associated with the intensity of their slow wave activity, such that regardless of whether they're young or old, the more intense their slow wave activity was, the better their overnight memory retention was as well. And this is similar to a lot of studies in younger adults, and then stimulation studies with enhanced slow waves and improved memory retention. And we did a mediation analysis with both age and prefrontal structure and found that the cortical volume was related to sleep expression, was related to memory, and that this actually statistically mediated the effect, such that the reason that cortical atrophy in the prefrontal cortex was associated with memory impairment was because of its disruption of slow wave activity itself. Now, I'm not saying that structural atrophy is always associated with slow wave activity. Other areas of generation are also more directly related to memory, like the hippocampus, for example. But sleep does play a role in this process and contribute. One of the questions we had is what is the reason that slow wave deficits related to memory impairment? We were showing that, in fact, older adults had sustained activation in the hippocampus following sleep when they were retrieving memories. And this was negatively associated with slow wave activity and memory, indicating there may be either a age-related deficit in memory consolidation itself. No, I'm sorry, I'm gonna stop. This is, give me a second. I need to turn off the rehearsed timings. This is not something I like. Okay, sorry about that. All right, let's continue from current side. Okay, great. Okay, that's better. So I'll go back a slide. Okay, there we go. All right. That's slide. Okay, sorry about that. All right. It could be that age is impacting the sleep-related consolidation process by disrupting slow wave activity, which then reduces the chance of memories to become more cortically dependent on retrieval following sleep. Another possibility, though, that's equally likely is that age is associated with disinhibition in the hippocampus and hyperexcitability, which then disrupts memory functions. We see this in MCI in early stages before you see a loss of hippocampal function. So these are both plausible explanations of these kinds of effects. And we see this both in our older adults and in those at risk for Alzheimer's, which I'll show later. In addition to loss of slow waves, we see loss of spindles, and one of the reasons why is because of degeneration of white matter. Others have shown that actually, even though they're not generated in the hippocampus, volumetric loss of hippocampus is also associated with loss of sleep spindles. That's Stuart Vogel's work in Canada. And surprisingly, thalamic changes don't seem to be robustly associated with spindles. That's probably because most of it's been focused on the entire thalamus and not really isolating the reticulonucleus function or structure. And so more studies are really needed to explore that contribution to spindle deficits with age. But the connectivity between different areas in the brain, those that connect cortical and subcortical areas in particular, show relations with changes in sleep spindles, particularly faster sleep spindles. And this is an example of that using DTI data where we had mean diffusivity associated with spindle expression. And then that was associated with white matter lesion volume, suggesting that it was actually white matter degeneration itself that was contributing to this change in white matter integrity that was impacting spindle expression. And there are functional consequences of these spindle deficits. One of the things we've shown is that memory encoding following sleep is disrupted on this base name task. And that's associated with reductions in hippocampus activity during encoding. So that means that age is associated with two deficits in hippocampus. One, increased activation during retrieval and decreased activation during encoding. And this may be shifting the balance between encoding and retrieval role of the hippocampus, which will impact the ability of the hippocampus to kind of be more able to just encode novel traces and lay them down as you experience new experiences. This could also be very relevant for the shy hypothesis, where you have this plasticity building up over the day. You don't have enough synaptic downscaling, and then LTP becomes occluded. And so, but this is also something that we see more associated with spindles. So there's a variety of different features of sleep that impact the way that the brain is supporting memory, which may have implications for some of the memory models. And we found, in fact, that hippocampus encoding activity and the memory function, or the face name memory during encoding, was directly associated with spindle density in young and older adults. We also found in a separate study that consolidation of a motor skill was also impacted by sleep spindle changes with age, but that was only if you were an older adult with a significant amount of white matter degeneration, then spindles were less able to support memory consolidation. And this is one of the things in the literature we've seen, is that in young adults, motor memory consolidation is supported by sleep and sleep spindles, but in older adults, we don't see it anymore. Well, it looks like it's not all older adults that lose this. It's when you have changes in the brain in particular, you have less robust sleep spindle dependent memory consolidation. So these spindle deficits are associated with both encoding of episodic and consolidation of motor memory. So you see impairments in both of these kinds of features. And the brain structure moderates this relationship, at least in motor memory. So it suggests that it's a complex relationship between how the brain is expressing sleep and how the infrastructure of the brain is able to support that expression of sleep. Okay. Another change with age that we've also been exploring is how the spindles and slow waves are coupled with each other. I'm going to show you different examples of how that changes with age using different approaches. One is an event-locked approach where we focus on slow wave events and look at how spindles are phase locked with them. And then we have a more kind of data-driven approach that we use later. In this context, we see that spindles and slow waves are coupled in both younger and older adults, but here's like a canonical slow oscillation. The red is young adults, the blue is older adults. And you can see there's a variability of phase of coupling in older adults that's much higher than young adults. And you can kind of see that here in these circular plots, which show vector length, which is represented as essentially the proportion of these spindle events that are essentially phase locked to a particular phase of the slow wave. And that's represented by the length of the line. The longer the line, the more likely that spindles are expressed at a particular phase of the slow wave. And here's showing the spindle activity as it goes up and down along the phase of the slow wave for older adults, and then for younger adults. And you see the younger adults are more phase locked. But the other aspect of this is that they're phase locked essentially at the depolarizing up peak in young adults, and they're closer to the hyperpolarized down state in older adults. So it's not just that there's more variability in coupling. It's not just that they're more likely to couple at a particular phase if you're young versus old. It's also when they couple that changes with age. We don't really understand fully why that's happening, but it is something that's been replicated by other studies. And here shows an example of that, where you see the power of spindles relative to these slow oscillations in older and younger adults. And you can see the depolarizing up peak of these waves here. The spindle expression is predominantly occurring there in young adults, but it's occurring slightly earlier, kind of in this period here, in older adults. And you can see sort of the length of the line. The vector length differs by age, particularly of our frontal cortex. And we also found that while the strength of coupling measured by this vector length was not significantly correlated, the phase of coupling was. So the more that you coupled to this depolarizing up phase, the better your memory was, the less your memory was impaired. Whereas if you didn't, the more you coupled predominantly away from that phase, the more your memory was impaired. So this age was affecting that. Here's an example of the comodulation index, or comodulogram, looking at modulation index, where we have more of a data-driven approach. We're just looking at as the slow wave or the carrying phase frequency here, and then the amplitude frequency on this axis, and the intensity of color shows the intensity of coupling. And in young adults, you see a predominant focused coupling, particularly in the slow oscillation frequency range, peaking across the spindle frequency range. In older adults, you see a similar thing, but it's much less dramatic. And when you compare the two significantly, you see there's a significant reduction in coupling in older adults relative to young adults. So age is impacting coupling strength, particularly in that frequency range where we see slow oscillations coupling with spindles. And we see there was a pretty specific frequency relationship with coupling here. Another way we can look at it is look like the consistency of the direction of phase locking, of the timing between slow wave and spindle events. Look at phase slope index, and in this case, if it's positive, that means the slow oscillation drives the spindle expression in coupling and leads it consistently. Whereas if it's negative, it means the spindle leads the slow wave, which isn't typically what you see in the data. And 0 means there isn't a clear, obvious relationship. And we see that there's this obvious relationship. The slow wave leads and drives the spindle expression in young adults, but in older adults, that directional relationship is impaired. And this is directly related to atrophy in the prefrontal cortex, which somehow must organize the coupling of these two. And in fact, in intracranial data, we know that the downstate of the slow wave induces the expression of spindles in the reticular nucleus. So atrophy of frontal areas that express slow oscillations may impact that relationship because they're disrupting connections between these brain areas. Okay, so if we think about these findings in aging, what does that tell us about sort of memory relationships? Well, we know if we look at the active systems consolidation model, we know that age is impacting the slow wave and spindle expression and their coupling. And this results and it is associated with impairments in all kinds of memory, including consolidation and encoding, and consolidation of both procedural and episodic. And so this tells us that these elements are really critical for memory. But in synaptic homeostasis hypothesis, we see a loss of slow waves. So this would suggest there might be a disruption in synaptic downscaling, and we do see deficits in encoding, though in our data we see it's more related to spindles rather than slow waves, which doesn't as strongly support this model. And finally, we also see that ages theoretically could be impacting the generation of new synapses because it's disrupting slow waves themselves and slow wave sleep expression. But this hasn't been as directly examined, so this needs to be kind of further examined. And the role of REM sleep changes in aging in memory consolidation really needs to be mined more. We just don't know a lot about that. We know that REM duration doesn't change all that much across normal aging, but REM expression does seem to, and so we need to that needs to be explored more, I think. Now I'm going to talk next and shift for the last part of the talk here and talk about sleep and its relation with Alzheimer's disease and Alzheimer's disease risk. Okay. So sleep showed profound, we've known this for decades and decades, profound deficits are seen in Alzheimer's disease here. And this is from a classic study from the 80s, where they looked at elderly controls versus mild, moderate, and severe Alzheimer's disease. And this is separated by women. We can see like one of the prominent changes is a loss of slow wave sleep, whereby the more severe dementia, the more severe the loss in slow wave sleep in both men and women. But the other thing that really stands out that's different from normal aging is a profound deficit in REM sleep that is seen. And this starts to occur even in mild stages of the disease that also progressively get worse across disease progression. And so REM deficits are really signature of dementia, I think, and it's seen in various forms of dementia, but Alzheimer's disease is one where you really see it. And this isn't just replaced with N2 or light stages of sleep. This is replaced with more fragmentation, more wake after sleep onset, more disrupted sleep. But largely this was thought of as an end stage effect of dementia ravaging your brain, and then your brain can't express sleep as well. But it turns out these sleep deficits also occur earlier in the stage. There's been a number of studies now, though they're smaller scale studies, a number of studies examining sleep in patients with mild cognitive impairment, specifically amnestic mild cognitive impairment. There's one study I was involved in that looked at this. And despite sort of matched time in bed, patients with amnestic MCI show reduced slow wave sleep, more fragmented slow wave sleep when it is expressed, reduced REM sleep, and increased wake after sleep onset. So this means that a lot of those global sleep architecture changes we see in Alzheimer's disease, we also see in mild cognitive impairment. So these sleep changes are occurring earlier in the process, not just at an end stage after neurodegeneration has essentially ravaged the entire brain. But the question then is, well, how early does this start? What are the mechanisms of these relationships? Well, in terms of how early it starts, there is now a lot of epidemiologic evidence suggesting that sleep actually increases the risk of Alzheimer's disease. Here's an example of one meta-analysis that I really like, which followed essentially or examined data from 18 longitudinal studies, nearly 250,000 people that were followed for almost 10 years, and then looked at various sleep disturbances in their relationship with different forms of dementia. And they showed that insomnia, symptoms, sleep disordered breathing, and just generalized sleep disturbance itself were all associated with Alzheimer's disease. But insomnia was more selectively associated with Alzheimer's disease, whereas sleep disordered breathing and generalized sleep disturbance were more general, impacting risk for all kinds of dementias, or at least vascular dementia and AD. And that makes sense for something like sleep apnea, right, where you have vascular, cardiovascular pathologies that are relevant for both dementias. Whereas insomnia doesn't have those same kinds of pathophysiological features. And this might mean that there are different mechanisms, depending on the type of sleep disturbance you see, that might be driving relationships with dementia and degeneration and cognitive decline. So this is something that I think will be needed to be examined in more detail in the literature as we go forward. In addition to increasing risk, sleep disturbance sleep also modifies other risk factors and their impact on dementia and cognitive decline. Here's a cumulative incidence of AD over 6 years on the top row. In ApoE4 negative and ApoE4 positive adults, you see dashed line is ApoE4 positive, solid is ApoE4 negative. And these are, this is the 10th, 50th, and 90th percentile of consolidation of sleep, the opposite of fragmentation. So people who have more fragmented sleep on ActiWatch, actigraphy-based data, these are the people with the least amount of fragmentation. And what you see is that AD incidence is definitely impacted by ApoE4 status, with the biggest genetic risk factor for sporadic AD. But that effect is greatly reduced if sleep is highly consolidated. And if you look at the global composite score for cognition and its decline over time, and those with the worst sleep versus the best consolidated sleep, you also see a similar relationship where cognition is compared and decline is exacerbated in people who are ApoE4 positive, but there's really no effect if they have very good sleep quality. So this suggests that maybe sleep may not only be in the path of the disease itself, but may also be a reserve factor or be associated with reserve factors in the brain. And so there's a lot of different ways in which sleep can interact along these different pathways. In terms of sort of mechanistic relationships, earlier studies also even examined earlier, what about pathological deposition in the brain? Is it associated with sleep in any way? And so Adam Spira was the kind of really the first to showcase this in humans, where they showed sort of amyloid deposition in their PET scans based on different subjectively reported sleep durations. And they showed that if you had greater than 7 hours or 6 to 7, you had less amyloid deposition than if you had less than 6. It was kind of a gradual increase in amyloid deposition the less and less you slept. And this was recently replicated in data from the A4 study by my colleague Joe Weiner, who essentially looked at a much larger sample and then looked at sleep durations and showed, in fact, that there was this effect, particularly at lower sleep durations, that there was, if you were sleeping less than 8 hours a night, subjectively reported, you had more and more amyloid deposition in the brain as measured through PET, global SUVR. There was a little bit of increase at 9, but it wasn't significantly different from 8 hours, probably because of smaller sample size. So there might be a U-shaped curve in this too, but we just don't know because we don't have the data that showcases that. The main effect we see is in short sleep associated with amyloid deposition. Tau has also been shown to be associated with various forms of sleep quality. Here's a measure from the medical outcomes study sleep questionnaire, where they looked at various sleep outcomes. The sleep adequacy is essentially, how restorative they felt in their sleep. And the more restorative, the lower the phosphorylated tau in their CSF. This suggests that, and total tau, phosphorylated tau and total tau. So this suggests that, in fact, sleep quality is related to that. In a study by in Holtzman's group, they showed that the more that sleep efficiency decreased over time in individuals, the more their tau changed in their CSF as well. And in this rodent study, they showed that chronic sleep restriction is actually leading to the phosphorylation of tau. So tau seems to be affected by sleep as well. And some classic studies from Holtzman's group in rodents show that there's an intimate relationship between sleep expression and sleep deprivation and A beta and tau proteins, such that on top you're seeing A beta proteins in interstitial fluid, and then the bottom you're seeing wake in minutes across these 3 days, and you see that there is a circadian rhythm, such that the more you're awake, the higher the A beta levels, the more you sleep, the lower the A beta levels. And this is also true with tau in their more recent study, where tau goes up during their active phase, and it's lower during their sleeping phase. And if you sleep deprive them during their sleeping phase, tau goes up. That's the red dots here. In addition, they also show that chronic deprivation increases plaque burden throughout different parts of the brain. And in addition to that, chronic restriction also encouraged the spread of tau between the hippocampus and the locus coeruleus. So this indicated that sleep deprivation was disrupting the brain's function and actually supporting increased pathological production, increased pathological plaque burden, increased spread of tau, as well as production of tau. Now, there's also been the other side of this whole lymphatic story, which I think is of great interest. And there's been a lot of sort of indirect evidence shown in rodents and in humans, very tantalizing. And this suggests that there may be a clearance function in the brain that essentially clears waste products and metabolites that is operated predominantly in sleep, that then may facilitate the reduction or clearance of these pathologies. And this started with Niedergard's data, of course, showing that extracellular space increases in flow and clearance of A beta was increased in sleep. And in addition to that, there's now some sort of tracer studies showing that tracer levels globally clear through the brain at a slower rate if you deprive people versus if you don't, particularly in areas that tend to accumulate pathologies like plaques. And then a nice, elegant recent study also from Laura Lewis's group also showed that there's flushing of CSF signal through the brain, which is associated with bold signal changes, but is preceded by and phase locked to increases in slow wave activity and delta power, and that they're linked to each other. So the idea was that the slow wave activity kind of precedes the brain CSF pulses and drives clearance through the brain. So it may not be non-REM sleep itself, but actually slow waves themselves that are operating this. But all this is indirect evidence. There is some controversy over the lymphatic system, what the biophysical properties and mechanisms are that make it work. But there is a lot of compelling data out there. And slow waves seem to be at the center of this story. Okay, so what about local features of sleep? Well, we see that early on in the process, and this is both in Alzheimer's disease, there are mild forms as well as mild cognitive impairment. You see loss of K complexes and slow waves, and you see losses of spindles, but those spindle losses are specific to faster frequency spindles, posterior sites, which is a little bit different than the age effect where you see deficits over frontal sites. This suggests there may be differential mechanisms driving these spindle changes, whether you have a dementia process versus a prototypical aging process. Excuse me. Okay. And one of the things that sort of got us thinking about this was that, the pathology, while it is global in many ways, especially at end stage, it starts out local. And the first places you start to see amyloid plaque burden and tau plaque and tau neurofibrillary tangle burden spreading to the cortex is really in areas of the brain that are known to be source generators, slow waves and slow oscillations. And so we thought, well, if pathology is particularly hitting these sleep generating areas, you're probably going to have very specific deficits in sleep expression. In addition to that, the medial temporal lobe, which supports ripple expression and somehow is involved indirectly in spindle expression, is also a site of neurofibrillary tangles early on, and also is a site of early age-related changes in neuronal loss. And in addition to that, we also see the cholinergic system, which supports EEG expression during REM sleep, also shows earlier degeneration and earlier tau deposition as well. And so this is sort of a question that we had is, well, maybe you have AD pathophysiology impacting the expression of oscillations that generate these different brain states and support the cognitive functions associated with them. And if that's the case, how early does it occur? Does it occur in MCI and AD? Does it occur in different stages of preclinical aspects of AD? And are there different consequences based on the changes of these different features? And so that's kind of the rest of the literature I'm going to focus on, is kind of trying to examine that question. And the first data is not ours. It's the data from Canada, where they show that there's a change in the way the brain expresses REM sleep in Alzheimer's disease. And what they did was they took, it's a fairly generalistic measure, but they took lower frequencies as a ratio with faster frequencies to look at how desynchronized the EEG was and how much slowing there was. The higher the number, the more slowing, because it's the lower frequencies over the higher frequencies. And they looked at various parts of the head, and they found indeed that AD patients had more slowing in their EEG and their REM sleep EEG, suggesting there was a loss of desynchrony mechanism. And this was, so different that they could fairly accurately separate Alzheimer's patients from controls just using this measure by itself. This doesn't just exist, however, in Alzheimer's disease. Another study that they published showed that, in fact, we see this effect in MCI, particularly amnestic MCI. It wasn't as obvious in non-amnestic MCI. So this suggests there might be a amnestic MCI specific change in sleep that may be heralding the onset of Alzheimer's disease based on a loss of REM desynchrony. And so one of the mechanisms that could be supporting this would be loss of the cholinergic system. And that was supported by this drug-related study where they used Nepazil, which is an anticholinesterase inhibitor, and said, what does that do to REM sleep EEG slowing? And in fact, what they found was in a placebo, there was no change in the slowing, in higher numbers, more slowing in the REM EEG. But over time on the drug, that slowing was reducing. So this indicated that, in fact, the change in the way the brain is able to express REM sleep EEG was, in fact, related to cholinergic expression. And they found some mild effects in cognition, but it wasn't clear it was related to the REM sleep change. In addition to that, there's a lot of rodent literature that shows bidirectional relationship between slow oscillation expression and amyloid pathology. Here's in APP expressing mice that rapidly produce amyloid, and they show deficits of slow oscillations as early as 3 months, which is 2 months before plaques, indicating that maybe the A beta proteins, not the plaques themselves, that are disrupting slow oscillations. They also took A beta media and they put it in wild types, and in fact showed that when they did the transgenic media, it reduced slow oscillation expression. If they synthetically produced A beta, it reduced slow oscillation expression. And when they optogenically manipulated mice in another study and doubled the frequency of slow oscillations, so they no longer had slow oscillations less than 1 hertz, they were all above 1 hertz, they saw actually an increase in amyloid plaque burden. And in a human study from Holtzman's group, they showed that the more they suppressed delta activity during sleep, the more the A beta changed in the CSF in humans. So this suggests that there's a manipulated bidirectional relationship between these two features. We've been able to show relationships in our studies whereby the more that slow wave activity is dominated by less than 1 hertz frequency range, the less amyloid accumulation there was in the brain, and the less it accumulated longitudinally over time as well. In Brandon Lucy's group, they showed that slow wave deficits were also related to tau deposition in the cortex. So once tau got to the cortex, probably likely mostly driven by his MCI patients, you then start to see slow wave deficits as well. We found there's a functional consequence of this, whereby the memory retention was reduced if you had less slow wave activity below 1 hertz. That's shown here. The more it's dominated by less 1 hertz range, the more memory retention there was. So this deficit associated with amyloid was relevant for memory. And recently, Brandon Lucy found that there was an effect on longitudinal cognitive decline over 4 years. That the higher their less 1 hertz slow wave activity was, towards a certain peak, that the more cognition was preserved, and the more it was reduced, the more there was cognitive decline. But he did find a U-shaped relationship, such that if it's over a certain peak amount, you also saw a decline. So this is something that needs to be explored more. But the bottom line is he found the significant association with less than 1 hertz activity, but not delta activity per se. There's also associations with tau and coupling between slow waves and spindles, as well as ripple expression. I'll go through this quickly because I want to get to the last study we recently published and finish up there. Essentially, the more the vector length, the more that the slow wave spindles were coupled with each other, the less tau there was in the medial temporal lobe. And there was really no obvious relation with amyloid, suggesting that tau was really driving deficits in coupling, and amyloid really wasn't affecting coupling between slow waves and spindles. In rodent studies, they found that ripple expression was disrupted in transgenics expressing tau, and that this was due to a disruption of inhibitory interneurons that delayed their expression and disrupted essentially their phase locking with slower frequencies. So this is the tau transgenic, and this is the wild type. You see there's a complete disruption of that, suggesting that there is a disruption in GABA neurons in the hippocampus due to tau, which is causing a disruption of it. Okay. So lastly, we had a study where we were trying to examine spindle expression and its relationship with a variety of AD-related factors, including AD-related proteins, but also inflammation, which is thought to be a core mechanism driving AD pathophysiology. And we found that there's a variety of different measures of inflammation, including GFAP, YKL-40, S100B, and soluble TREM2, which all showed associations with spindle expression over frontal, central, and even in parietal, some cases, the spindle expression. Now, what you see here, if it's a white asterisk, it's corrected significance across the scalp. If it's a blue dot, it's significant uncorrected. And if it's a green asterisk, it means it overlaps with a bunch of different measures that we examined. So there's a lot of consistency with effects where the more activation, and these are measures that are good measures of activation of microglia and astrocytes in the brain. And the higher they were, the more activation of these glial features, the more deficits and spindles that we saw. We also saw deficits associated with AD-related proteins, particularly tau, and measures of synaptic and axonal degeneration, NFL, neurogranin, and alpha-synuclein in this case. And this is flipped because it's a reciprocal. It's essentially the more of these features, the more phosphorylated, more total tau, more NFL, more log neurogranin, and the more alpha-synuclein, the lower spindle expression. And we found indeed that that was associated with deficits in memory, whereby the more the spindles showed deficits in these areas, the more memory was impaired. And there was overlap in all of these measures here, showing there's a very consistent effect. And so we then did a mediation model where we tried to examine how are all these features related to each other. And we found that there were a couple of different significant paths. One through YKL40, which impacted tau and alpha-synuclein, which had a downstream impact on spindle expression. And we also found another path through soluble TREM2, which had a significant effect on tau and alpha-synuclein, particularly phosphorylated tau, which had a downstream impact on spindle expression. And these were really specific to YKL and TREM. The GFAP and S100 effects weren't nearly as robust. They weren't there significantly statistically. But we also found these associations were there for spindles, for when we use spindle algorithms. And we also found other trends through other features, but these are really the main paths driving it, suggesting that it might be that age is impacting microglia activation in particular, which is affecting synaptic integrity and phosphorylation of tau, which is having a downstream impact on spindle expression. We also were trying to examine whether or not this was specific to oscillatory activity itself or the aperiodic activity that might be changed by the EEG. And so what we do is we take the EEG power spectrum and we take it apart. We model out the 1/f, and we model out the oscillatory activity, and then we model out sort of the offset, and we compare those features. And what we found was that really this effect was really driven not by the 1/f, which is more related to excitatory and inhibitory balance and neural noise, and not the offset, which is related to spiking activity, but the oscillatory activity itself. And that's shown here as an example where phosphorylated tau and alpha-synuclein relationships are really significant with the oscillatory component of fast sigma activity and not the 1/f exponent itself per se. Lastly, we have some preliminary data that's not published that we tried to look at coupling and how that might be related to different features. So we looked at modulation index, and then we looked at using a comodulogram approach where the peak of modulation index was in the slow wave frequency space and where the peak frequency of modulation index was in the spindle frequency space. What we were seeing actually was this, it matters what frequency that spindles are coupling with slow waves. And if they're faster, you tend to have better memory. And it turns out that a lot of these features are impacting that expression such that the more phosphorylated tau, the more NFL, the more alpha-synuclein, and the more activation of glia, including microglia, the slower that spindle frequency was over these sites. So this suggests that coupling and the features of slow wave spindles are being altered by pathophysiological features of Alzheimer's disease. And this has theoretically a downstream impact on memory, but we're exploring that. So finally, just to kind of conclude and finish up, so we have a couple minutes of questions. I'm sorry, it took a little too long. If we look at these different memory theories, we see from these data from the literature that A beta is associated with disruptions of slow oscillations specifically, which seems to be associated with worse sleep-dependent consolidation. Tau disrupts pretty much everything else, the coupling between slow waves and spindles, ripple expression, and spindles themselves. And this effect seems to be enhanced by CNS inflammation, disrupting tau, disrupting these features. And this seems to be resultant in memory impairment. The synaptic homeostasis hypothesis, based on the findings, we would expect that then if A beta is disrupting slow oscillation expression, but not delta expression, then maybe there would be hyper downscaling and enhanced active forgetting, but this is something that we need to examine in future studies. Whereas once tau hits the cortex and all slow waves are disrupted, we might start seeing something similar to what we see in aging, which is more just hypo downscaling and just reduced occluded LTP and learning ability. In terms of this multi-state consolidation, based on the effects of Aβ and tau on non-REM expression, you'd think that would disrupt the ability to form new synapses, whereby disruptions associated with cholinergic degeneration might disrupt the theta and the synchrony-related mechanisms might support spine elimination and stabilization. But this needs to be explored in future studies. But I think a lot of the findings we have are really kind of consistent with the predictions of the actin system consolidation hypothesis. Okay, and so that's all I have to start with. Thank you for your time. This is, of course, studies that were supported by NIH funding, but also through collaborations with people at Wisconsin and my mentors, Ruth Benka and Barb Benlin, as well as my former mentors, Matt Walker and Bill Jagist at Berkeley, and collaborations throughout the world in different places. And a lot of these analyses were really kind of spearheaded by my graduate student, Abhishek, as well as one of my junior specialists, Kitty, and a lot of our work is ongoing in the lab. But thank you for your time. And I'm happy to stay on as long as you want to take questions. And I'm sorry that I kept you